You ran DISUC or Wilcoxon on your SCORNA-seq data. You have 500 differentially expressed genes. Now what? Reading each gene's function one by one would take weeks and miss the big picture. The library catalog organizes books by section. Gene ontology and pathway databases organize genes by function. Enrichment analysis asks, are your 500 genes disproportionately from one section of the catalog? 500 genes. Some are kinases. Some are cytokines. Some are metabolic enzymes. Some have unknown function. The pattern is invisible when you look at them one at a time. But if 40 of your 500 genes belong to the inflammatory response pathway and only 20 were expected by chance, information is overrepresented in your gene list. This statistical overrepresentation turns a list of genes into a biological hypothesis. Gene ontology organizes biological knowledge into 3 hierarchies. Biological process, what the gene does at the system level, immune response, cell cycle, apoptosis. Molecular function, what it does biochemically, kinase activity, DNA binding, cytokine receptor binding. Cellular component, where it acts, nucleus, membrane, extracellular space. Go terms are nested in a directed acyclic graph. Inflammatory response is a child of immune response, which is a child of response to stimulus. Enrichment at a specific term is more informative than at a broad parent term. Overrepresentation analysis builds a 2x2 table for each pathway. Row 1, genes in your list that are in the pathway and genes in your list that are not in the pathway. Row 2, total genes in the pathway that are not in your list, and total genes not in either. Fisher's exact test, or equivalently the hypergeometric test from the probability garden, computes the probability of seeing this many or more pathway genes in your list by chance. Test every pathway. Adjust for multiple testing with Benjamini-Hochberg. Significant pathways are overrepresented. Multiple databases organize genes differently. KEGG curates metabolic and signaling pathways with beautiful hand-drawn diagrams showing the molecular interactions. Reactome focuses on biochemical reactions and events with more granular detail. MSigDB, the molecular signatures database, organizes gene sets into collections. Hallmark sets distill complex biology into 50 well-defined signatures for cancer and immune processes. Use KEGG for metabolism, Reactome for signaling detail, and MSigDB hallmarks for high-level biological themes. ORA has a fundamental limitation. It requires a binary cutoff. A gene is either differentially expressed or it isn't. Move the threshold from adjusted p less than 0.05 to 0.01 and you might lose half your John list and have your enrichment results change. ORA also ignores the magnitude of change. A gene with log2 fold change of 0.3 just barely passing the threshold counts the same as a gene with log2 fold change of 5. Gene set enrichment analysis fixes both problems by using the full ranked list. Gene ontology organizes biological knowledge. ORA tests for overrepresentation using Fisher's exact test. KEGG, Reactome, and MSigDB provide curated pathway databases, but ORA depends on an arbitrary cutoff and ignores fold change magnitude. Next, GSEApy eliminates both limitations.